Good evening to you all. Let's confirm that you can see my screen and you can hear my voice. So if you can hear me and you can see my screen, kindly put up your hand. Let's confirm that I'm audible and the screen is also visible. Okay, great. So a number of you have confirmed that. That's good. Okay, so we can continue. It's exactly 8 o'clock over here. Your time is 5 o'clock. So we start now, and then we hope that the others will also join quickly so that we can finish early. In the last lecture, I think we finished with the structure of nucleic acids. So as I told you that, I mean, biochemistry at all times, you realize that it's structured in such a way that you take a component and you study the structure of it, what it is made up of. And then after that, you begin to look at the functions. So we started looking at nucleic acids. We finished with the structure of nucleic acids. Now we are moving into the function of nucleic acids. And what basically is the function of nucleic acids for the storage and transfer of genetic information. And how does a nucleic acid do that? The transfer of the information. So we will look at this function as what we refer to as gene expression or protein synthesis, because as a matter of fact, all what it is that from the DNA, we're looking at the overview of it at the end of the last lecture, that from DNA you have a process by which messenger RNA is synthesized, and then from the messenger RNA you have proteins synthesized. But today's lecture will be in such a way that a lot of the things we'll have to watch them first as video before, like some animations, before we begin to, talk about it, because it's one of those things that is just difficult to imagine if you are just looking at it on paper, and if you are just listening to somebody, talking about it, it's difficult to comprehend. So we'll look at some animations, and then after the lecture, I will see how best I can send these animations in addition to the presentation to you, so that you can watch those animations even before you go reading your textbooks and using the presentation as a guide to do that. I will strongly recommend that you watch the animations before you even go reading, because it can be difficult if you are just looking at it on paper. Okay, so before we even start, let's start first of all to remind ourselves of what the nucleic acid, DNA, RNA, the structure that we looked at. I want us to first of all look at an animation of that. That's where we'll start from, and then from there we will continue to look at the processes, the functions that we're talking about. Okay, so let's take the first first animation, see how best we can make some sense out of it.
This video we will study about the nucleic acids. Nucleic acids are basically biopolymers which are made up of monomeric units known as nucleotides. We have two important functions for nucleic acids, that is they hold our genetic information and they perform variety of another functions that we will discuss later. We have two important types of nucleic acids in our body, the DNA and the RNA. Let's first try to understand the definition of nucleic acids. So nucleic acids are biopolymers. Bio means that they are synthesized inside biological systems and they are not synthetically synthesized. They are polymers, which means they are made up of repeated units called as monomers, which in this case are nucleotides. Now take this example. We have many different types of biopolymers in our body. One of them are the proteins. And most of you know that the proteins are made up of repeated units called as amino acids. Similarly, nucleic acids are also important biopolymers in our body, which are made up of repeated units, which are known as nucleotides. So the next important thing is to understand the basic structure of a nucleotide, which will help us to understand the structure of nucleic acids in detail. Now this is a simple diagram explaining the structure of a nucleotide. As you can see, the single nucleotide is made up of three important groups, the phosphate, the sugar, and the nitrogenous base. And these groups are attached to one another by bonds, which we'll discuss later. To understand the complete structure of a nucleotide, let's try to understand the structure of these groups one by one. So the first is the sugar. The sugar present inside the nucleic acids are 5 carbon sugars. Now most of you know the 6 carbon sugar present inside the body, and most important of them is the glucose. Now in chemical nomenclature, all the 6 carbon sugars are called as hexoses. The word hex meaning 6. There are different types of hexoses in our body other than glucose, like the mannose and galactose. Similarly, 5 carbon sugars are called pentoses, the word pent meaning 5. There are different type of pentoses in our body, but the two important type of pentoses that are present inside the nucleic acids are the ribose and the deoxyribose. So this is a simplified structure of the sugar present inside the nucleotide. Now if we compare the structure with the chemical structure of the ribose and deoxyribose on the right, you can see the structure clearly. We have carbon atoms on each of these corners. Note that the last carbon atom is located outside the ring. We have an oxygen atom at the center topmost position. You can also see in this chemical structure that the carbon atoms are named from 1 to 5 in a clockwise direction. So the carbon atom next right to the oxygen is called C1. So C2, C3, C4 and C5. C5 is located outside the ring. You can also see in this chemical structure we have different type of functional groups attached to these carbon atoms like the hydroxyl group and the hydrogen atom. The most important of these functional groups which determine whether the sugar is ribose or deoxyribose are present at the carbon number 2 and the carbon number 3. In the sugar ribose, we have a hydroxyl group that is attached to the carbon number 2. This is the chemical structure of ribose. Now, if you compare this structure with the structure of deoxyribose, the only major difference is at carbon number 2, that the carbon number 2 contains only a hydrogen atom and not the hydroxyl group. So the resultant sugar is called a deoxyribose. Here you can see the carbon number 2 in deoxyribose contains only a hydrogen atom, whereas in ribose, the carbon number 2 contains a hydroxyl group. You can also tell this by the difference in their names that the deoxyribose has a prefix called as deoxy, which means removal of oxygen. The resultant sugar, the deoxyribose, is more stable as compared to ribose because of one less functional group. The deoxyribose is present in DNA and ribose is present in RNA. You can also remember this by the starting of the DNA, which starts with D, so the deoxyribose, and the RNA, which starts with R, so the sugar is the ribose. The next important group to study in the structure of nucleotide is the phosphate group, which is this entity right here. The phosphate group consists of a phosphorus atom in the center to which 4 oxygen atoms are attached which are negatively charged. The phosphate group is the same phosphate group which is present in the adenosine triphosphate, the energy carrying molecule of the body. However, in ATP, we have 3 molecules of phosphate which are attached to one another by high energy phosphodiester bonds. The phosphate group is a polar molecule due to the presence of highly ionized oxygen atoms which impart negative charge to the phosphate group. Now, the third important structure present in the structure of a nucleotide is the nitrogenous base. Nitrogenous bases are basically molecules that contain nitrogen in varying amounts and they act as a base. Now, most of you know that the human body contains organic chemicals which have carbon, hydrogen, and oxygen in varying amounts. In nitrogenous bases, nitrogen combines with all of these atoms to form ring-like structures. These molecules are called bases since they can donate electrons to other molecules and form new molecules in this process. The nitrogen combines with all of these other atoms to form ringed structures. Now we can have two different types of ringed structures. One where there are single rings and second where there are double rings. The single rings are called pyrimidines and the double rings are called purines. We have three different types of pyrimidines, the thymine, cytosine, and the uracil. And we have two different types of purines called adenine and guanine. In the center, you can see the chemical structure of each of these bases. As you can see, the purines, which are double ring structures, adenine and guanine. Due to the presence of double rings, these purines are larger as compared to the pyrimidines. The thymine, cytosine, and uracil all are single ring structures and they are smaller as compared to the purines. The difference in the size of these bases is very important as it helps to pair these bases in the structure of DNA properly, which we'll study later. So now you know the individual structures which are present in a single nucleotide. Next, we will look at how these individual structures bond to each other to form a single nucleotide. And then we will look at how many nucleotides bind to each other to form a chain of nucleotides, which is called a polynucleotide, which can be either DNA or the RNA. So if we take a sugar and name just the important carbons here, which will be the carbon number 1 and the carbon number 5. And of course, you can see that at the carbon number 2, there is only a single hydrogen atom. So this sugar will be a deoxyribose. Now to pair this deoxyribose sugar with this pyrimidine ring, the bond formed will be between the N1 of the pyrimidine ring and the C1 of the sugar. If you want to go into the details about how these nitrogens are named, you can check the link in the description below for the IUPAC nomenclature of the aromatic compounds. Now this bond will be an example of a glycosidic bond, which is a covalent bond which links carbohydrates with other structures. Now if we have to pair a purine ring at a similar position, the bond formed will be between the N9 of the purine ring. Now next, let's attach the phosphate. The phosphate group attaches with the carbon number 5 of the sugar and the bond formed is the example of ester bond, which is also a strong covalent bond. So now you know how the three structures in a single nucleotide bind to each other to form one nucleotide. Next, if we look at how many nucleotides bind to each other to form a chain of nucleotides called polynucleotides. So you can see basically what happens is that the phosphate group that attaches to the carbon number 5 of the sugar below also forms a bond with the carbon number 3 of the sugar above. And this pattern is repeated again and again. So you can see that the phosphate groups forms bonds both above and below the chain of these nucleotides. So as a result, a long chain of nucleotides is formed, which consists of a sugar phosphate backbone. And in the center, we have all these nitrogenous bases which project from the sugar phosphate backbone. Now, this can be an example of RNA if the sugar present in this example will be a ribose sugar. But if we take a similar strand of polynucleotide and run it in opposite direction, and this strand binds with the original strand through hydrogen bonds. Now here comes the role of complementary base pairing. Now, what complementary base pairing says that only adenine forms a hydrogen bond with thymine and only guanine forms a hydrogen bond with cytosine. Now, this is a universal rule that is applied in the structure of a DNA. So here you can see adenine forms a hydrogen bond with thymine, and similarly thymine forms hydrogen bond with only adenine. In similar case, guanine forms a hydrogen bond with cytosine and vice versa. Now this is the basic structure of DNA, and you can compare this with the step ladder structure of the DNA. You can see that the supports of the ladder in the red are basically representing the sugar phosphate backbone, and the rungs of the ladder represent the nitrogenous bases that are forming hydrogen bonds with each other. Now of course, the original structure of DNA is that of a double helix. So you can compare this structure with a double helix, where the helices of this structure represent the sugar phosphate backbone, and in the center you can see the red lines which represent the different nitrogenous bases that are forming bonds with each other. So this was an introductory video explaining you the structure of nucleic acids. In subsequent videos, we will study in detail the structure of DNA, the structure of RNA, DNA replication, transcription, translation, and many.
All right, so that was an animation on nucleic acid structure as we study. I hope that gives you a good summary of what we did so far on the nucleic acids and a good revision. Now, we want to continue to look at the function of the nucleic acids. And as we started looking at it previously, we were looking at the overview, the summary of it. We talk about the processes of replication, transcription, and translation. Now, before we go on to look at the process of replication, we'll first look at an animation on replication, and then we go on to the slide to look at replication. So let's move on to look at replication.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, so that was
DNA is a molecule.
That was DNA replication that we have seen in an animation. So we will continue to look at the process of DNA replication on a slide. So which is the beginning of the function of the nucleic acids, DNA replication, where we just saw the formation of a new DNA molecule from an old one. So what we saw was referred to as a replication fork. And that is what we are seeing on the slide here, the replication fork. Now you can imagine if you are just looking at this on a slide, how difficult it can be if you hadn't seen the animation before this. So that's why I was advising from the beginning that, in fact, in learning these things, you have to use animations, use the videos to understand the processes first of all, before you start looking at it on paper. Because on paper, it can be so hard to understand. So what you see is that, first of all, you saw an enzyme referred to as helicase or primase, which was responsible for unwinding the DNA strand. So as it unwinds it, so you have a DNA gyrase also. Okay, actually the DNA gyrase is what is turning around to unwind, and you have the DNA helicase or primase sort of supporting that DNA molecule that has been, ripped open up, because of course, before you can have say the formation of a new strand on the old strand, there should be the opening up of these hydrogen bonds that are between the nitrogenous bases. And what you saw was that there was one of the strands being referred to as a leading strand. So here, the old strands are the green ones. So the leading strand is the green one, which is at the top, and the lagging strand is the green one, which is below at the down. Now on each of these, you have new nucleotides coming to be synthesizing a new strand that is binding onto that old strand. Now, one thing which you have to keep in mind is that the synthesis of the nucleic acid strands are always in the direction of the 5' end to the 3' end. So you see that the orange strand, which is the new strand, which is being synthesized, you see it's being synthesized from the 5' end, which means the old strand on which it is being synthesized, it starts from its 3' end. And so you see that strand, the orange strand, which is the new one, getting synthesized on that leading strand continuously. But on the lagging strand, because the formation of the nucleic acid strand is always from the 5' end to the 3' end, that lagging strand is in the opposite direction, which means the synthesis on it cannot be continuous. So the synthesis is done in what is being referred to as Okazaki fragments. So these are small chunks of nucleic acid strands that are being synthesized onto that lagging strand. Some other things you saw was like, the enzyme, the DNA polymerase, which is responsible for bonding these new nucleotides onto the old nucleotides to form the new strand on the old strand, to form a new DNA molecule. You also saw the DNA ligase, which came at the end on the lagging strand, because the lagging strand was forming in chunks. There will be some spaces in between them. And ligases are enzymes that are responsible for joining these nucleotide molecules that have gaps in between them. It's a form of finishing the process of the synthesis of the nucleic acid. So you see at the end of it that the molecule, that like you have a DNA molecule out of which you are having 2 molecules of DNA, and each of the DNA molecules that were synthesized as new ones have been referred to as semi-conservative, or if the process itself, the process of DNA replication, is referred to as a conservative process, because the new strands which are being synthesized will contain one old strand. I mean, the new DNA molecules which are being synthesized will contain one old strand and one new strand. So you have an old strand which is conserved and a new strand bonded onto it to form a new molecule. So you have a semi-conservative process. All right, so let me see. Already, yes, when we are doing these things, I always will know that, yes, there should be questions, because things can get, not so clear. So now, Sanja Godwin is saying, please, during the replication, I can see that the new strand is straight, but later it forms wave-like structure. What initiates such wave-like structure? Oh, okay. Godwin, it isn't that when the synthesis was ongoing, like say natural, if like the synthesis is ongoing, the strand is going to be straight like as you are seeing it, but then later it becomes wavy. No, it's just that because it's an animation, it had been done like simplified such that when you watch it, you will understand it. That's why you see that. But it's not like first the thing will be straight and then later it becomes wavy like that. It's just that at the end they were showing the double helix structure as how the nucleic acid, the DNA structure looks like. So that is why it's like that. It's an animation of a complex process which has been simplified. That's why you saw it that way. Okay, I hope that's clear. All right, so I'm monitoring the chat. So if you have any questions, anything, any difficulty, just write in the chat and then I will attend to it. Or if you like to speak to it, means that you have to note it down what you want to say, and then at the end you can raise your hand. I will enable your mic, then you unmute yourself and you speak. Then I can respond. All right, so we'll move on to the next process. The next process. Here too, before we move on and start looking at, all the details of transcription, I will want us to again watch an animation, and then we continue from there. Yeah. I hope that's come. All right.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription unit. This portion of the DNA will be used to produce RNA. Upstream of the transcription unit is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TBP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP into ADP and PI. RNA polymerase then synthesizes an RNA
All right. It appears I went off for some few seconds. Okay, so did you see everything till the end before there was that short break? I hope you saw everything before there was that short break. But whatever it is, I will share the videos with you so you can watch them individually as well. So that was also process of transcription in animation. So transcription, which is the process of the production of a messenger RNA in the nucleus from the DNA. So we see the process, first of all, starts with the opening up of the DNA molecule, because the base is a particular fragment, which is a gene coding for a particular protein, had to be activated, opened up, and be exposed so that that information can be copied onto a messenger RNA. And here, the enzyme which is responsible for that process is the RNA polymerase. We see the strand which is being copied is referred to as a template strand. And it is from the template strand that you have the messenger RNA copied from. And you see, take note that here again, the synthesis of the messenger RNA is from the 3' end to the 5' end, from the 3' end to the 5' end. What did we see with the synthesis of DNA? When we're looking at DNA replication, you see that the synthesis is from the 5' end to the 3' end, from the 5' end to the 3' end. Here, well, in a way, yes, actually the synthesis starts from the beginning, which is the 5' end this way. So from the 5' end towards the 3' end. So it's the same direction in which the synthesis of DNA was occurring, that you see the synthesis of the messenger RNA also occurring from the 5' end towards the 3' end, because it's going in that direction whilst it is moving away gradually from the template strand. Now, this process is in 3 stages: initiation, elongation, and termination. So I think we'll look at another process of transcription in animation. So I think before we do that, let's just look at these processes first of all, transcription, and then we can go and look at the other animation before we move on to translation, because I think that other animation will be combining transcription and translation together to give you that full process of protein synthesis or gene expression. So we see in the DNA strand, there are portions that are referred to as promoter regions or recognition sites. And I think you saw that in the animation being referred to as the Tata box. And that is particularly for eukaryotic cells. I mean, the Tata box only means that you are having this repeated sequence of thymine adenine in those regions. And those regions are actually, recognized by, say, DNA polymerase, or let's say the RNA polymerase, because this is about the synthesis of RNA from DNA. So the RNA polymerase recognizes this site. So you see the RNA polymerase coming to bind onto that promoter region. So that's a form of activation of the process. So the initiation, the process can then start. And once the RNA polymerase binds, you now see that there is that opening up, the ripping open of the DNA molecule ready to be copied onto the messenger RNA. Now, then we see there is the process of elongation. So from initiation, where you have the RNA polymerase binding onto the DNA molecule, there will then be the start of the nucleotides that are complementary to the nucleotides of the template strand, now coming together in that complex of the RNA polymerase and the DNA template strand, and be joining and be forming the messenger RNA. And you see the direction is from the 5' end to the 3' end, just like you saw with the DNA replication. And one thing you should take note of is that in DNA replication, because it's a DNA molecule that is being synthesized, it means that the complementary base that will come, against adenine will be thymine. But in case of RNA, you take note that the base which will be coming there will be uracil. So because that's a messenger RNA that is being synthesized, so the base there is going to be uracil in place of thymine. All right, so Dorcas Akwete says, please, is the Tata box usually at the middle of the DNA, or it can be anywhere on the DNA? Okay, that's a very good question, Dorcas. So when you take a DNA molecule, there are portions of the DNA molecule that code for specific proteins. And that's what we refer to as gene. So a gene is a particular fragment of a DNA molecule, or a particular region of a DNA molecule that is coding for a specific protein. So for instance, is that particular region we are talking about coding for, let's say, salivary amylase, which is a protein? Or is it coding for the alpha proteins of hemoglobin? Or is it coding for another maybe enzyme like pepsin? So it can be coding for different things. Or is it coding for the hormone insulin, for instance, which is also another protein? So different regions on a DNA molecule code for different specific proteins, and those regions we refer to as genes. So do you have a gene that is coding for different proteins that will be functioning in different processes? Now, at the beginning of every gene, you have the recognition sites, which you have the Tata box as a component of. So the Tata box is at the beginning of a gene. So the RNA polymerase recognizes that and goes to bind onto it. Then that will be the initiation of transcription. So then messenger RNA begins to be transcribed from that particular region, that gene, before the messenger RNA will then continue the process, go to the cytoplasm for further processes. Dorcas, I hope that is clear. All right. Then, okay, good. All right, so after you go through that process of elongation, of course, the process has to come to an end. So, and that's what we refer to as termination. So we have a termination of transcription. So there too, you have the same way as we have the regions which are promoters like the Tata box. We also have some regions that, because it's a gene, it has a certain length, and when it gets to its end, then the process stops. And what we are seeing here is an example of what we saw with the structure of, I think when we were looking at the secondary structure of RNAs, we saw the hairpin structure. So here you are seeing a function of that hairpin structure, where at the end of a transcription process, a hairpin is formed. It's like the messenger RNA molecule is getting zipped off, because the moment it comes to bind on like these complementary bases bind to each other that way, then it zips off. It gets hydrolyzed off the DNA template. So that's a function of that hairpin structure. That's a process by which you have a termination of transcription. So then you can have your messenger RNA then going into the cytoplasm now. So carrying the message to the cytoplasm. And once it gets to the cytoplasm, the message will be translated. So the next stage that we'll move to will be translation. So before we move on to that process of translation, let's see one last animation. This one is combining both the transcription and the translation together to give us the full process of gene expression, or what we refer to as protein synthesis. Then from there, we'll just come and finish it up with the slides.
In order for our bodies to function.
So, let's see. Okay, showing now.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes, transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliciosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down 1 codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body to occur properly, such as breaking down food particles in digestion, and the processes of transcription and translation make the production of proteins possible. In order for our bodies to function, we need to supply.
All right, so that was the process of transcription and translation combined to give us that whole process of gene expression. So when we refer to gene expression, what actually we're just referring to is the synthesis of proteins out of the genes, because the genes are coding for specific proteins, and these proteins have specific functions in the body. So that is the genetic information that is being stored in the DNA, and which will then first have to be copied onto the messenger RNA, and then the messenger RNA brings the message from the nucleus to the cytoplasm, and in the cytoplasm you have the process of translation to have the protein synthesized out of it. So we're seeing the start of the process of translation, initiation, where you see the RNA strand, that is the messenger RNA strand, and the start codon is being shown. Previously, we saw the genetic code, and I mentioned to you that just keep in mind the short form of August, AUG. That is what codes for the amino acid methionine. And so methionine is the first amino acid that starts the process of protein synthesis. And so you see, onto the start codon, you are seeing a small ribosomal subunit combined to a transfer RNA. And that transfer RNA is having what is being referred to as the anticodon. It is that anticodon that is going to bind to the start codon. So the anticodon is complementary to the start codon. And the amino acid that is bonded onto it is methionine. That is represented there as MET, methionine. And once it binds onto that messenger RNA, the process of translation is initiated when you now have the large ribosomal units, the bigger green parts that comes to bind onto that complex to give you what is referred to as the initiation complex. So now the full complex that initiates the process of translation is formed. That is what is being referred to as the initiation complex. Now let's zoom in into the initiation complex. Let's zoom in into the initiation complex. So the initiation complex, particularly the large ribosomal units, is having three distinct sites that are referred to as the E site, the P site, and the A site. I mean, E refers to exit, because that is where the transfer RNA that comes into the complex with an amino acid to bind onto the rest of the amino acids to form a protein chain. That's where it leaves the complex. So it's referred to as the exit site. So when we watched the animation just before this, we saw that process where you saw the transfer RNA leaving the complex from the E site, the exit site. The P site also means the peptidyl site, because that is where like you are finding the peptide being formed. And the A site is the amino acid site, because that is where the amino acid which is being carried on the transfer RNA is coming into the complex. So you have these three distinct sites in the initiation complex. All right, let me take a question. It says, please, I know of transcription and translation of prokaryotes and eukaryotes. Please, I want to ask whether we are interested in the eukaryotes or both prokaryotes and eukaryotes, replication, transcription, and translation. Okay, so particularly what we are referring to now is for eukaryotes. So this is for eukaryotes. Right after this, we'll touch a bit onto the process in prokaryotes. What you find, say, in bacteria, some difference that we'll touch on. All right. So, by what we've been looking at has been with. So this is like the translation initiation in prokaryotes. You find that the first of all, the promoter region. One of the differences you find is that we refer to the Tata box in eukaryotes. In prokaryotes, what you find as the promoter region is what is referred to as the Shine-Dalgano sequence. So that's different. It's not the repeating sequence of thymine, adenine, thymine, adenine, but you see this sequence of guanine, guanine, adenine, guanine, guanine as the Shine-Dalgano sequence, which is that promoter region in prokaryotes. So that's one difference. Then another thing is that in eukaryotes, I mentioned that the amino acid, which is the initiation amino acid, that is the amino acid which starts the process of translation in eukaryotes, was methionine. In bacteria, it is a modified form of methionine, formyl methionine. So there is a derivative of methionine as the start amino acid. The start codon is the same, but the amino acid there is a derivative of methionine. There is some formyl group that is attached on that methionine. So that's a major difference that you'll find in prokaryotes. But the process generally is the same with the ribosomal subunits, the small subunits, the large subunits, and the sites that are in the large ribosomal subunits are the same as you can see over here. All right. So I hope that's okay with Raymond, who was asking about the prokaryotes and the eukaryotes. Now, there has to be elongation. We saw initiation, but that's just one amino acid which came into the complex methionine. After that, there should be more amino acids that will come into the complex and bind onto the amino acid, the first one. Then the third amino acid will come, the fourth, on and on until you have a long peptide chain. And all the time, so this is, if you see, this is a cycle of the elongation process where you have a transfer RNA, which is carrying an amino acid, and that transfer RNA comes into the A site. And once it comes into the A site, the next thing is that you'll find that there is a transfer of the amino acid which was on the transfer RNA in the P site transferred onto that amino acid which is on the transfer RNA which has just come into the A site. I mean, that will be by an enzyme. Some peptidyl transferase is responsible for such transfer of amino acid from the P site onto the A site. So even if a chain is forming, the chain will be moving and be joining the next amino acid. So what it means is that the first amino acid which came in will always be the beginning. So that methionine will always be the beginning of the peptide chain which is being formed. And I think actually that's the methionine is what will give you the N-terminus. That is that end of the amine group of the peptide chain which will be forming. Methionine will be that N-terminus. So whichever amino acid is coming in the last will give you the C-terminus of the peptide chain which is being formed. So the process continues until you see the once there is a transfer of the amino acid or say a peptide chain onto the new one, there is a shift of the complex on the messenger RNA. And when that shift occurs, it means the A site becomes empty, whilst you see that the empty transfer RNA, I mean the transfer RNA which has been freed from the P site when there was a transfer of an amino acid or a peptide from it onto the next amino acid, that moves to the exit site, the E site, and then leaves the complex. So the process goes on and on in that cycle as the process of elongation. Now, we have come to the end of the process of translation, where once you reach the end, that is, you have hit a stop codon. Remember when we were looking at the genetic code, there were some stop codons. And what were they? I think we saw UGA, UAG, and UAA as the stop codons. So those codes do not code for any amino acid. So when it gets to that, you have reached the stop codon. So we see on the stop codon, no amino acid is carried by a transfer RNA to come onto that complex for the process to continue. But what you have is what is being referred to as a release factor. So a release factor is a protein. It's also a protein. So these are proteins that sort of comes to give a signal or just block the process from continuing. So it comes to bind onto that stop codon. It's not like it's some nitrogenous bases that are coming to bind on that stop codon, but because how it is depicted, you might think that it's some nitrogenous bases that are binding onto the stop codon, but actually it's a protein that just comes to block that site, and so the process doesn't continue again. And it actually acts as an inhibitor to the peptidyl transferase, the enzyme which is responsible for the transfer of like the peptide which is forming to be joined to the next amino acid. So what it does is that once it inhibits that peptidyl transferase, that process will not continue. And at the same time, it also activates, the process of hydrolysis of the peptide which is being formed to be, released from the transfer RNA. You see, there's a last transfer RNA which is in the P site. So that then will then get hydrolyzed and leaves the complex. And at that moment, you can have the whole complex dissociating. Then they can go into the process of synthesis of another molecule, of another protein molecule. So you have a free polypeptide, which is being released. The ribosomal, both the small subunits, the large subunits, and even that release factor, the messenger RNA itself, they all dissociate into their single components. And so they can form other complexes again, and then they start the process of, the synthesis of another protein molecule. So this brings us to the end of that process of translation, transcription, translation, what generally we refer to as protein synthesis. I know this is a complex topic, and so as I was saying from the beginning, I will advise that you use the videos to have some understanding of the process before you start looking at it in black and white on paper, because when you are just looking at it on paper, it can be difficult to comprehend step by step how these things occur. So the floor is open. You have any question, anything? I'm happy at least today you have been more active. You've been writing in the chat, asking questions along the line. I hope you will continue to be interacting like that all the time. I noticed that the evening group is more active than your group. They are always asking questions, contributing, and they write in the chats, they talk. So I wish you also do same. All right. So I will stop my talking here, and then I will open the floor. Any questions, any contributions, any concerns?